Hello students, today we shall learn the concept of prevention and regulation of cancer. As we are all aware that there is a saying, prevention is better than cure. It holds good even in the prevention and regulation of cancer. The prevention and regulation of cancer includes the first step, namely is to keep the identification of carcinogens and later keep the patient away from the exposure of these carcinogens. Example, tobacco smoke, frequent X-rays and UV ray exposures. Second is AMS test. It is a test subjecting patients to get the idea of mutagens and cancer. It is a screening method used to identify the potentiality of carcinogens that acts as mutagens. In simple, it is the ability of mutagens to induce cancer can be easily identified by AMS test. In this method, bacterial strains that has mutations upon exposure to these chemical mutagens are cultured on a medium with histidine. So bacterial culture medium contains histidine. So bacterial strains forms the colony without depending on the histidine as it has mutations. Hence, number of colonies indicates the mutagenic potency of a chemical or a mutagen to be tested. So, AMS test is to test the mutagenic potency and cancerogenic effect of a particular chemical. The third important method is change of lifestyle. Change of lifestyle will be an effective treatment in case of cancer. There are few methods that are used to treat or to kill the cancerous cells. The first method is radiotherapy or radiation therapy. In this method, high energy particles or waves are used to kill cancerous cells. Cancerous cells upon exposure to these radiations cause damage in DNA that stops functions such as cell growth and division. It ultimately results in the shrinkage of tumors. It is a localized therapy used to cure or reduce early stage of cancer. Also prevents the returning of cancer in case of patients. Example, X-radiations are used to treat the cancer types such as prostate cancer, skin cancer, head and neck cancer, throat cancer, larynx cancer, breast cancer, brain cancer, colorectal cancer, bone cancer and ovarian cancer. The radiotherapy is achieved by three methods: external radiation therapy, internal radiation therapy, systemic radiation therapy. In external radiation therapy, the high energy rays are delivered to the cancerous tumors by means of machines. In internal radiation therapy, a radioactive seed or pellet is used inside the body and thereby being very close to the tumor, thereby radiations emitted by these radioactive pellets will frequently kill the cancerous cells which are nearby located. This method is also called as brachytherapy, B-R-A-C-H-Y, brachytherapy. The third method is systemic method of radiation therapy. In this method, introduction of radioactive chemicals are introduced into the body either through the mouth or through the blood vessels. So radioactive chemicals are introduced into the body of the patient. So these radioactive chemicals are chemicals which are equivalent to the radioactive compounds, and these chemicals are called as radiomimetic agents. Such radiomimetic agents are used in systemic type of radiation therapy. Coming on to the advantages. Large proportion of cancerous cells are killed in this method. Second, it helps in shrinkage of tumors. Third, relatively it is a safe method which is painless. Fourth, cancerous organs need not be removed or by preserved through the method of surgery. Coming on to the disadvantages, it causes damage to the neighboring cells also other than the target cells when we subject it for radiation process. The second step is in disadvantage, poor healing of wounds is noticed. The third point is there is inconvenience of radiation therapy dosage. Mean to say 5 days a week, sometimes 6 days a week, sometimes alternative days of a week are used in radiation therapy. The last point is it induces medical disorders because it also affects non-target cells.